Okay, I'm backpedaling just a little bit just to show you the slide we looked at before, where we're looking at a dendrite on one side and a synapse, or a couple of synapses here with axon terminals and vesicles, just because it's a cool electron micrograph of this area. We looked at how some of the effects of how to clear the synapse. Here we can see the life cycle of a neurotransmitter, so we understand that the neurotransmitters first has to get synthesized, it has to get stored, and here we are in the presynaptic area. It has to get activated at the end of an action potential by that voltage gate for calcium, which will allow calcium to come in, activate vesicles, where they are moved to the membrane here at the very end, and then the exocytosis. And once that happens, they move over and so here's the release, and here's the reception on these post-synaptic receptors. And this is our next topic, is to talk a little more about that. Postsynaptic receptors, once they've docked and opened some ion channels or perhaps activated something internally, then the neurotransmitter either diffuses away, gets broken down by enzymes and catabolism, or is brought back into the axon terminal by reuptake, sometimes by specific transporters that will work that way. And the one picture that's not in here is the role of the astrocytes. This is where we look at the astrocytes. So this idea that there is something called a tripartite synapse. There you can see that 3. So it's kind of like 3 parts, tripartite synapse. We see that normal axon to dendrite here, where that activity is shown, but that here, hanging around, is a process, meaning just like an extension off of an astrocyte that has these different receptors itself, which is able then to take in some of these materials that the neuron has released into the synaptic cleft, and can then do various things with them. And what we're seeing more particularly, because this is what we've talked about so far, is this idea that there's some recycling by the astrocyte. So it's part of the clearing of the synapse mechanism for some neurotransmitters. Is this astrocyte is also helping by taking in, see, it has a receptor for it, taking in that neurotransmitter, changing it, reconverting it to something, and then sending it back to this presynaptic neuron through some kind of a transporter system. So that's what you see here. And they're telling us here that this happens to be an mGlu-R, or a particular type of glutamate receptor. And we're going to look at some others too. So here's a cool picture, because this is an actual electron micrograph. You can see here's the dendritic spine here. The synapse would be somewhere right in here. Here's that presynaptic neuron, that's its axon that you see there. And then there's the astrocyte, kind of right around there. We mentioned last time too that we typically see this kind of activity. So here's the neuron. You have to think sort of upside down, because this is kind of opposite the way we typically look at it. But here's the soma, the cell body of this neuron, and here's its axon, and it has its synapse right here on the dendrite. So that's the axo, meaning axon, dendritic synapse. But there are other options. There can be synapses right here on the cell body. And so that means that there has to be some way for reception of a neurotransmitter to happen here. And then, really interestingly, we have this axonic activity. So here you see 3 neurons, these 2 here. This one is having an axosomatic synapse right there, but now this one is synapsing on the axon of this one. So stay tuned, we'll talk about that. this is where we look at the interesting part, really, of how this electrochemical signaling happens. So neurotransmitters, you've chosen, many of you already have chosen your research on one of them. And we know that they are made in the neuron, and then they are used as ligands on the neighboring neurons for communication. So that's one thing, but importantly, in some sense, are the receptors, because it's the receptors that are going to determine what kind of responses are going to happen. So we have two types: ionotropic, fast ones, open channels, ion channels, and metabotropic, slower, activating G protein systems, which then can do a variety of things. And then, as many of you are going to see very soon, certain neurotransmitters have certain receptors that are called receptor subtypes. We'll see some examples coming up. Already talked about this. I elaborated a little more than this picture is showing. We have mentioned, or maybe we just stopped there and now we're mentioning it here, we know what a ligand is. When a ligand binds to a receptor, it can activate it, and something that activates the receptor, meaning that it can be even a different chemical, something that activates a receptor, which causes the normal activities to happen, is called an agonist. An antagonist is something that's going to block it. And an inverse agonist is going to actually cause what you'd think would be the normal response of that receptor. But in fact, in other words, it does what the agonist would normally do, except the effect is opposite of that normal function. So agonist is the normal effect. Antagonist is something that's going to block, and an inverse agonist is something that it activates, but in an inverse way, an opposite way. We're going to look at neuropharmacology. Remember, we talked about that a bit, and like, what are drugs, what are chemicals, what are medicines that we take in that can act as any of these three? And if they can come in and basically do something at that receptor at the normal area that a ligand would bind, that's going to be a competitive ligand. Now, you remember this, I think, from biology when we talked about enzymes, and we talked about how enzymes can have competitive inhibition when something goes directly to its active site and kind of interferes with that particular enzyme's active site. So that's competitive. Same areas being affected. A non-competitive ligand is sometimes called a neuromodulator. And these, similar to, if you remember, allosteric inhibition, but this isn't allosteric inhibition. It's similar, but not exactly like it. But it's, in this case, a ligand that binds to some other site, some site on that receptor, that will change the way the receptor can interact with a neurotransmitter. So we'll get into a few specifics on this as we go. We're looking again at drugs here. So we're talking about, well, how would this affect a normal activity if it's an agonist or an antagonist? And I've talked about this a little bit in the last video. So just take a good look at the pictures and see if it makes sense. Why it's so important to understand this area here is that all of this is really a target for drugs. So it could be, illegal drugs or medical drugs, and anything that would be considered a neurotoxin. It'd be this area that we'd be most concerned with, because this is where the message is getting passed on and that activation or that inhibition can happen here. As an example, if a drug or a neurotoxin interferes with the synthesis, in other words, the making of a neurotransmitter in that presynaptic cell, that could be an issue, right? So if we're going to target the synthesis, what could be targeted, the drug could do a couple things, and it might interfere with some of the enzyme activity necessary to make it, for the synthesis process. Could be that a drug is targeting the ability for the reuptake process to happen, and it could be in the vesicles themselves. Could be that the drug would target the release in the first place, or somehow in that fusion and the recycling process. Could be that the drug, its job or its effect is to do something to the neurotransmitter once it's in the synapse, block its reuptake, inhibit the way it's broken down. And then another possibility is that the drug is designed to block the neurotransmitter from binding in the first place. Now, this part is what you're going to be doing a lot on, so I'm going to really just give a couple of examples here. This first example is to look at glutamate and to say, well, yeah, glutamate, we know what it is. We know that it's an excitatory neurotransmitter. As a matter of fact, the most excitatory one we know. And glutamate can activate one of both of these types of channels. So it can activate ion channels. So it has ionotropic receptors, or it can activate a G protein coupled receptor. And just to show you, and you don't need to know all of this, but just to show you that the different flavors of these are the subgroups of receptors that would be glutamate related. So if it's a fast-acting ionotropic glutamate receptor, these all qualify for one of those types, something called NMDA, something called AMPA, something called kainate. All of these will do the same thing. They're fast, excitatory ion channels. But they have some little differences because you say, well, why should we even have these different ones? And we'll talk a little bit about that. Here on this one, on this AMPA, ionotropic AMPA receptor for glutamate, you see a GLU R1, GLU R2, GLU R3, and GLU R4. R means receptor, and this is just telling us here's some subtypes of this receptor. We go over here, metabotropic receptors, they have group 1, 2, and 3 with their own, notice the M in front of it, that's the metabotropic part of that. This one is slow but excitatory. These are slow and they inhibit. So remember, this is where it gets a little bit crazy because we said glutamate's this excitatory neurotransmitter, but now we're saying, yeah, but now we have some receptors though that are going to cause some other action to happen. In just a minute, I will go through just a small little snippet on how neurotransmitters are made in the first place, starting with a group that are of a big category, if you remember, called biogenic amines. And under that umbrella is a subgroup called the catecholamines. And these are the examples: dopamine, norepinephrine, and epinephrine. So this, why it's called a catecholamine is because it has this element to it. It's got this thing called catechol, which you may have seen something like this before. Here's those OH groups, those hydroxyl groups. And this is a carbon ring. And this, when it forms a catecholamine, here's the nitrogen part, remember amine here. And this all doesn't matter at all. So it's just to show you that there's the chemical structure for all you chem nerds out there. Don't get worried on this one, because somebody's going to talk about this anyway in their presentations. But here you see that how these different neurotransmitters are made actually can be shown by this pathway. And the first part of the pathway is this amino acid called tyrosine. Tyrosine you get in your diet. And so this is the first step. You have to have tyrosine in your diet in order for tyrosine then gets converted into something called L-DOPA. And L-DOPA gets converted into dopamine. And dopamine then can get converted into norepinephrine. And norepinephrine can be converted into epinephrine. And what you see here on the sides are the different enzymes that are necessary for those conversions. So remember, we said if we were going to target something, if we wanted to target the synthesis of something or to slow something down, we could try to target one of these enzymes that would affect the rest of the pathway. It's just to show you that the catecholamines get made into dopamine, norepinephrine, and epinephrine. So I think I'll stop there because I don't want to steal anyone's thunder, but I'll talk a little bit more about this later as to how we could see this whole process in a particular area of the brain, and we'll talk about that soon. So stay tuned.